where your registered address is. You know, if you changed your driver's license to Pennsylvania because you moved here for school, or if you are still registered at home in your home state, I need you to get the absentee ballot. I need you to know how to do it and where to do it and how to get it and how to send it in and when the deadline is. All of that stuff you guys need to figure out now while there's still a year left to do it, so that in that year you don't have to be rushed or feel like you made a mistake or, you know, find out later that it isn't going to work. We're going to do it right now so that everybody who's listening to this channel, like literally everybody who's listening to this channel, needs to register to vote. Oh, hello, Frank. So you got another really hard paper here again. It's a hard paper that requires an extraordinary amount of background knowledge to figure it out. And that background knowledge has to do with the learning processes underlying addiction. It has to do with silent synapses and their presence during development and their presence during learning. And a new hypothesis about how silent synapses might contribute to cocaine craving incubation. I mean, it's an incredible amount of background information that not everybody's going to have. And so let's just start somewhere with that. And that is that the nucleus accumbens is involved in cocaine seeking and cocaine addiction, and changes in the nucleus accumbens have been associated with cocaine seeking behavior. Interesting observation is that AMPA receptor complements change in the nucleus accumbens as the behavioral output in response to cocaine or after cocaine. As the behavior of the animal changes, AMPA receptor complements change within the nucleus accumbens. So what this group is going to try to do is dissect this circuit a little bit and look at one specific excitatory input known to be important. This person just crossing on the light. Known to be important in addiction, and that is a basolateral amygdala projection to the nucleus accumbens. And they're going to use a similar technique that was used in Henri Oswald's paper. They're going to label a specific set of neurons with optogenetics so that they can target their axons with blue light and stimulate them to release glutamate. And then they're going to use this ability to target this specific connection to look at how this connection may or may not change during various time periods during cocaine withdrawal, relapse, cocaine seeking behavior due to withdrawal. And the observation that was made in the past was that there were silent synapses. Hypothesis theory was thrown forward that this AMPA receptor component change that occurs many days after the last time you were exposed to cocaine has to do with this incubation period and how you tend to seek, the animals would tend to seek cocaine, not the day after they had it, but many days after. And at 45 days after, there's a very strong increase in this cocaine seeking behavior. And so they're trying to figure out if there's a synaptic component to this, if there's some change that occurs in this time scale that could help to explain the synaptic underpinnings of this incubation of cocaine seeking behavior. And this is on the background of the idea that AMPA receptor complements change in the nucleus accumbens during this period when the animal's behavior changes. It goes from an early withdrawal to late withdrawal, an increase in the seeking behavior. And that's a pretty cool phenomenon that should have a synaptic component. If what we believe about synapses is true, then there should be some component like this. And that's what's beautiful about this paper, is that figure by figure they're just going to set this up and dissect this circuit, looking at this one component of the behavior that they're looking at, one aspect of it, and that is the presence of silent synapses or not. And presence of silent synapses could indicate a learning event. And so the idea would be that silent synapses have NMDA receptors but not AMPA receptors, and AMPA receptors are very fast, high-speed sodium channels that are activated by glutamate. There are other glutamate receptors in the brain, of course. Those are NMDA receptors that we're mentioning here. Two major categories of glutamate receptors. The difference being that AMPA receptors are activated at hyperpolarizing potentials, and they have extremely high conductance and fast open and close times. I don't know if they use the word desensitization for AMPA receptors, but they inactivate, such that they open, they let a lot of sodium through in a temporally tight window, and then they close. The slower component of synapses is the NMDA receptor. And the NMDA receptor is unique because it's blocked by magnesium at resting membrane potentials. So only when the neuron is active or when there are enough AMPA channels depolarizing in the local region of the cell will the magnesium block be relieved, and then NMDA channels can also conduct sodium. But in addition, they also conduct calcium, and as we talked about last week and the week before, calcium runs the show. Calcium causes changes to occur, like proteins to insert, genes to be transcribed, trafficking of different proteins occurs. What is this person doing? I don't know. But so NMDA channels tend to be thought of as a sort of plasticity cue. When NMDA channels are open, synaptic plasticity can occur and other things can happen because calcium comes in. And so a silent synapse identified during development and such, that was also a Pittsburgh left. Did you see that? During development and such, these are thought to be synapses composed only of NMDA receptors. And the consequences of this functionally are that at rest, if glutamate gets released and binds to only NMDA receptors, then at that particular synaptic contact, if you're recording in that cell, you won't see anything because there won't be any current. The magnesium block won't be relieved, and so NMDA receptors won't open, and therefore you won't see a depolarization. So the synapse doesn't do anything at regular resting membrane potentials. So a silent synapse needs depolarization in order to be active, and then the idea would be that if there are silent synapses present and there's glutamate released on those silent synapses during synchronous behavior in the neuron, then they'll relieve the magnesium block, and the relief of the magnesium block could cause plasticity at the silent synapse, synaptic plasticity being the insertion of AMPA receptors, and voila, you go from having a silent synapse to having a mature synapse. So what they want to, their hypothesis is in this paper then, that cocaine seeking, the incubation of cocaine seeking behavior after cocaine withdrawal is hypothesized to occur by the generation of silent synapses by the exposure of cocaine. They've shown this with previous papers. And these silent synapses then, over the course of the 45 days of withdrawal, start to mature, because calcium permeable AMPA receptors insert to complement the NMDA receptors. And that insertion then would be blamed for the increased cocaine seeking behavior that doesn't occur when the synapses are still just silent synapses, or just NMDA synapses, but occurs only when those synapses mature over the course of the withdrawal period and then generate the behavior. So in figure 1, they're just showing you that they can do the experiment that they're planning to do, which is they're going to inject channel rhodopsin in the basolateral amygdala and get it expressing there. And then after they get it expressing there, they cut slices and they look at the axons which are labeled in the nucleus accumbens shell. Those locations in the slices are shown in that figure. And then when they stimulate with blue light, they can evoke synaptic responses that are presumed to be from the axons labeled by the injection in the basolateral amygdala. So just like the Oswald lab paper, we're labeling something functionally selected as relevant. In the Oswald paper, they were just labeling inhibitory neurons. And in this one, they're going to label excitatory projections from the basolateral amygdala. And those excitatory projections go lots of places, but one of the places they go is the nucleus accumbens shell. And this location will become relevant when they stimulate it in vivo. This is also a Pittsburgh left. Just watch, here he goes. He's going to do it. You can hear him revving. See, there he goes. That's a Pittsburgh left. So, in figure 2 then, what they're going to do is take that same connection and now try to demonstrate to you what the plan is. So, in figure 2, A through D, I think, A through D are behavioral experiments where they just show you how they train the animal and how the nose poking behavior changes over the course of the learning. And then in that figure is where you really need to see that at day 45, the cocaine seeking behavior in the cocaine exposed animals is much higher than those exposed to saline. And that's different than day 1, and I think that's what's cool about it, is it's really a thing that happens over the course of the month you haven't had cocaine if you're a rat. So E through M are the slice experiments where they cut the animal at a particular time point and then stimulate the fibers with a blue light and measure the responses that they see. And now this is where it becomes tricky, because here's where they're going to quantify, here's where they quantify the presence of silent synapses or not. And if you look at the raw data that they show in E through H, or whatever it is, E through L, I don't know what it is, can't remember anymore. But they show you cocaine animals and they show you saline animals. They show you the responses evoked at -70 and the responses evoked at +50. And remember again, the reason why they're doing that is because the peak at -70 is mostly AMPA, because NMDA channels don't open because there's some magnesium block at -70. And although the peak current at +50 is a combination of AMPA and NMDA, we know from looking at the -70 trace and from the exhaustive knowledge we have about the kinetics of AMPA receptors that by 50 milliseconds or so, they're all closed. So if we just look at the peak at, say, 50 milliseconds after stimulation in the plus 50 millivolt condition, we're really just looking at NMDA channels. And so what they do here is an interesting assay that takes a little while to understand, but they titrate the light to get some failure rate. And then once they've titrated the light to get a certain kind of failure rate, then they leave it there and they compare everything across the different groups. The tricky thing for me is M, because I'm not, the formula that leads up to how they calculate the percentage of silent synapses in my mind is a pretty impressive formula, that it's definitely not a formula that you just drop out of a hat, because it's one of those formulas that, you know, it's not A plus B divided by something. It's a weird natural log that I think has precedent from another paper, but because the results that they show, I don't see usually when you look at the raw data, you can see the effect. Your brain can recognize it, and then the statistics that are done in the bar graphs just convince you of it. But it's not immediately clear to me, in that figure, in those traces, where they show the trials from 1 to 100 with the stimulation of the axons in those different conditions, it's not immediately clear to me the trend that they show in M. M makes it seem like, well, duh, there's mostly silent synapses in the cocaine group, and they're only there on day 1. But if you look at the raw data, you don't really see that at all. And I'm not sure if it's because I'm not looking at it right, or if I just don't see it, because it's a subtle effect that only comes out when you do their gymnastic math. But I'm not telling you that there's a mistake here. I'm just saying that this is one of the places where I feel like we need to read more or discuss more with the author about how they justify this calculation, because the bar graph that is shown in 2M is not obviously drawn from the data shown in the previous panels. So for me, it's not really clear the effect that they say that they're calculating, and I understand that it's there, and in the materials and methods you can read their calculation, but to me, I just like data where it's like, you know, the difference is this mean and that mean, and you know, it's obvious. And to me, it's not immediately obvious in the raw data what the effect is that they then show so clearly in M. That's my only beef. Hope that makes sense. Walking on his phone. Still didn't move. Far nugget. So in figure 3, they introduced this calcium permeable AMPA receptor selective antagonist. So it's only blocking AMPA receptors that are permeable to calcium. That's not all of them. And specifically the one that they believe is inserted due to other data from other labs is inserted during this change over the 45 days of withdrawal. And in figure 3, they're just going to look at day 45 only, and they're going to show, and this is kind of a, well, for me on the bike it's a hard figure to explain, but let's just say that figure 3 now is going to show you that specifically calcium permeable AMPA receptors seem to be responsible for this increase in the component that they see the change, and it doesn't occur, this insertion does not occur in the saline animals. And so that's cool, but it sounds like the reviewers didn't like that enough. So the reviewers maybe asked for a day 10. So figure 4 is a similar analysis where they used the blocker to block that selective component and then see how the total signal changes. And then from that change, they can make some estimation about how much, what proportion of these AMPA receptors are calcium permeable. And so in figure 4, they insert an extra time point here. They insert a 10-day time point, which is kind of developmentally in terms of how the learning occurs or how this cocaine craving incubates over time during the withdrawal. Then it's not clear to me why we're stopped, but then there should be some incremental change at day 10 that sort of indicates that, okay, we started with silent synapses only at day 1, and then there's some transition which occurs to day 45 where all those silent synapses, or a lot of them, have matured and collected calcium permeable AMPA receptors and could underlie the incubation of cocaine craving. So now it gets a little more complicated because, again, it's failure rate and it's stimulation at this minimum threshold frequency that gets them this weird, not weird, but gets them this ability to come up with a semi-quantitative way of measuring the silent synapses and the amount of AMPA receptors that have inserted. And so in figure 5, what they're going to look at specifically is to try and show you that the NMDA component has not changed, but the AMPA receptor component has changed. And this would again jive with previous data that's shown that in other ways, but this would be the first time where now we're stimulating a functional set of synapses, a functionally isolated set of synapses, and showing that those synapses in particular, remember these are amygdala synapses inside the nucleus accumbens shell. So that's pretty sweet. And so this is now starting to close all the holes and really make it a pretty solid observation. We've now got a time course where more or less their hypothesis is confirmed, and now they do specifically look at rectification, and rectification gives them measurements at different voltages. I don't know anymore which one had the rectification figure in it, but it's important to understand that an AMPA component that doesn't have calcium permeable AMPA receptors in it will basically have an IV curve that is linear. So the more negative you go, the more current will flow. The more positive you go, the more current will flow. And that slope should probably be linear for the most part, and it is for a lot of general ion channels. It's only dependent on driving force. But with calcium permeable AMPA receptors and a lot of potassium channels and other channels that show selectivity, they don't conduct the same in both directions. And in this case, there's an inward rectification here where positive currents, but conductance at positive voltages is reduced in comparison to the same on the other side of the axes. And so you might want to get a little better definition from the author on inward rectification and outward rectification and how they're using that. But now it gets very cool, because in figure 6, they're going to look at an LTD protocol that has in the past been shown to be able to change things behaviorally in the rat or in animals. So if we induce an LTD protocol by stimulating the basolateral amygdala, or is it the nucleus accumbens? That's an interesting one. You might want to figure that one out. What the other papers are showing, because I didn't look those all up. But essentially, they reference papers that have shown that LTD can change the behavior, cocaine-seeking behavior. So what they're going to do now is try to induce LTD in this pathway using their optogenetic tools and see if stimulating this pathway with a similar LTD inducing protocol, that they reverse the synaptic change that they see at 45 days, where first they see silent synapses at day 1, and over the next month they mature by adding AMPA receptors. So the question becomes then, can they induce LTD and effectively measure these calcium permeable AMPA receptors being withdrawn from the synapse? And these last two figures are, of course, what made this a Nature Neuroscience paper. So here they do it in slice first in this figure 6. And then they use similar measuring tricks in whole cell at different voltages so that they can tease out the different components, AMPA and NMDA, and then they use that naphthalm or whatever it's called, 5-letter, starts with N, only one vowel. Use that calcium permeable AMPA receptor selective antagonist, use that drug to show that the only, or very likely in silent synapses, that these things are changing because the NMDA signal doesn't. Only the AMPA channels and only the calcium permeable AMPA receptor component changes, and only in cocaine treated animals. So of course the obvious checkmate then would be to do the same stimulation protocol in a live animal that you expect to see cocaine, looking at figure 2 again, A through N, A through D, where they show the basic behavior. And so what you'd expect then is that if they stimulate LTD in the saline animal, it won't matter. In the sham animal, it won't matter. But in the animal that received cocaine, they should start seeking on the day 45 test. And if they don't, after LTD, then you got all sides of the puzzle. And indeed, that's what they found. The animals with optogenetically induced LTD connection between induced in the connection between BLA and nucleus accumbens show greatly reduced nose poking behavior at day 45. Checkmate. So that is really a pretty cool paper, and it's spectacular that it's at Pitt. Yavin Shaham, I don't say his name right, he's like a monster at NIDA, National Institute of Drug Abuse. That guy basically is a demigod of drug abuse and NIH funding. And he's like third author on the back of this paper. So they were connected with good people, and if Yavin says this is good, chances are pretty good it's good. So I hope you get a lot out of this paper. The discussion will probably be pretty lively. And I hope this abbreviated single ride will be a little bit easier and useful too. I don't think I'm going to edit the morning ride, although I might. I'll line it up and see what comes out. So in case you don't know, the Amazon rainforest as a whole is on fire. It burns every year, but not this much. Incidentally, also, Siberia and the northern tundra of Canada is on fire. Large uninhabited places are burning. And at the same time, Donald Trump wants to release 9 million acres of untouched forest from Alaska because, you know, we don't need forest and climate change is a hoax. We need wood. We need wood more than we need forest. So anyway, try to stay alive out there. Don't let all the contradictory evidence get to you. Don't let all the contradiction get to you. Don't let all the counterintelligence get to you. Trying to break us down, tire us out, and make us stop paying attention. And a lot of us have. But we, the young people, need to be awake. And even if the old people are just going to stay in their own stupid ways, we need desperately to wake up and register to vote. Make sure you know where your polling place is. Double check that you're registered for real. And then already now schedule the day off. You're taking election day off this year. Everybody is. That's the way we're going to win, is when we have plenty of time to wait in line. When Dunkin' Donuts gives free donuts away after you vote. When you get a free hamburger from Burger King when you vote, or free Pepsi. Be well, guys.